Walters is a bioengineer working at the nexus of synthetic biology, cell engineering, immunology, and cell therapy. His current research focuses on ways to make cell therapies more effective and accessible for deployment in regenerative medicine to address both systemic and neuroinflammation. He recently completed his PhD in bioengineering, jointly advised by Dr. Caleb Basher at Rice University and Dr. Scott Olson at UT Health Houston McGovern Medical School. He is now a postdoctoral research fellow at UT Health, working to bring the therapies that he designed during his PhD to the clinic. Welcome.
Ready. I'm a little bit taller, so I don't know if the microphone's picking me up okay. But I'm really excited to be here. As she said, my name is AJ, and I'm going to be presenting a lot of work that was done jointly between Dr. Olson and Dr. Basher and I during my PhD, and that now I've moved over into my postdoc. So this is several years worth of work going into this. So the starting position that we want to get into is that I work with mesenchymal stem or stromal cells. They're very useful. They're immunomodulatory. They have those regenerative properties. Importantly, we're able to manufacture them, which will be a key point for my talk. They are able to be genetically engineered, but that's historically been a challenge in having them being engineerable and manufacturable. And importantly, they're also immune evasive and safe for allogeneic use, which makes them a viable candidate for off-the-shelf therapy. They've been used for that with the landmark approval at the end of last year of Ryoncil, which was the first MSC approved product for acute graft versus host disease in children. The major challenges that still face these therapies and why it took decades to get them into an approved a therapeutic approval is that they have several problems, some of which work together, some of which fight each other, but they have been faced with insufficient potency or incomplete understanding of their mechanism of action, poor targeting, limited persistence in the patient. I'm working on all of these characteristics, but today I'm just going to talk about their potency and how we can build them so that we're not relying on their native therapeutic action. Instead, we take advantage of their safety, but build in all of the therapeutic effect in the engineered function so that it's reliable regardless of donor and manufacturability variants. So one of the cool things is in our group, we are part of I call it a translational triangle that goes all the way from research and translation through, we have our own GMP facility, all the way to we have clinical partners that run these clinical trials with these MSCs. So the clinical team and the GMP manufacturing team informed what we needed to match as we were engineering these cells and helped us come up with this clinical manufacturing constraint framework. Essentially, we need to get the product over this orange line before it crosses this gray line. So we have to make sure we hit ideally up in this top section of the plot. That allows us to hit the clinical scale without losing this manufacturability. So as simple as that ended up being, as it seems, it ended up being one of the major challenges of my PhD to overcome. We thought we'd knock it out in like 6 months, and it took several years. The reason for that is there's two common error modes that many of you might be familiar with, is that in error mode C, we see this reduced proliferative capacity that causes the cells to senesce as they take on this transgene. And then the other option is that the engineering is so toxic that it drops and never recovers through this giant bottleneck that needs to happen through selection. Just so y'all are aware, I'm also going to use this pie chart representation to show the data where the orange line of the circle matches this clinical scale, and an ideal product would be mostly, if not all the way green and larger than the orange circle or the orange dotted line. So as I mentioned, after lots of time and most of the data that I'm not showing you here, we were able to get that idealized expansion compared to this wild type. So we were able to get well above the orange line, or an order of magnitude above it. The product matched exactly the parameters that we wanted. And then the replicates, it was important, it was reproducible. And we only had to select for one passage. And then after that, the transgene was stable, which is pretty atypical compared to other engineering approaches that we've worked with in the past. Usually you have to keep the selection on through the entire expansion. The important thing here is that this manufacturing process did not affect the potency of the cells. Both the wild type and the engineered cells behaved similarly in our potency assays, which means now we have the ability to build on new features into these cells without having to compensate for any damage that we introduced during the manufacturing process. So what we wanted to do next, I work with a lot of synthetic biology tools, which are generally encoded in large DNA constructs, but they provide all kinds of cool new features, whether that's multi-gene cargos to deliver five drug products at once, or being able to do sense and response circuits or small molecule inducible circuits. So we wanted to know what the limit was of how far we could go. So in this case, this is what that same product looks like. So this is the basic cargo, and we have a little over 10 to 20 fold over that orange line that we can work with. When I increase the size, this is where the GFP, BFP, mCherry, and iRFP oh my came from, which is that I added an expression unit in order to increase the size of the cargo with independent promoters for each of these genes. And we saw that at selection, which is this passage 3, there's a significant drop in yield from this cargo, but then after that, they expand at the same slope, the same wild type-like expansion. You can see in these pie charts here that there's a very significant difference in the yield that you get from those products, as well as the homogeneity as you increase with genes. But what we did after that was I switched it, instead of using genes to expand the size, I expanded the size using non-coding DNA spacers. And we completely lost the effect of on the expansion and retained quite a bit of the homogeneity. So it was all coming from the expression burden, not necessarily the DNA size, which you would expect with this system. So looking more closely at the flow cytometry of this, we were able to also see that we're losing a lot of homogeneity in the product as we're increasing. So these first 4 constructs are the single gene arrays of all the different colors, and we get nice homogeneous products for each of these. Then when I have 2 genes, or 3, including pure myosin, which obviously isn't a fluorescent, we still have nice homogeneity, but then when I go to 3, and then 4 fluorescent proteins, we lose a lot of that homogeneity in the product. So looking just at the GFP, which is the one that we're making the decisions based off of, you can again see how stark that is as you go from 3 genes to 4 genes to 5 genes. Comparing that to the same size cargos using the spacers, that we're not really losing any product characteristics that we would want. So what this tells us is that we can build out these constructs in large DNA cargos as long as we tune the expression level down of different parts. Or what we then did was making an inducible system so that the transgene is off until you want it on during the manufacturing process. So this is a relatively simple genetic circuit. In this abstraction, the system acts like a video game controller where you add the inducer and you only get the transgene on demand. The important thing about this configuration is that it uses human-derived proteins, and it uses an FDA-approved small molecule inducer. So it's more translatable than other systems that have been used in the past. It's not one that we necessarily invented, but it is one that my colleagues in the Basha lab spent a significant amount of time learning the system and optimizing it. But it's not something that we had used in a therapeutic context. So that's where I stepped in, is how do I build this platform into a therapeutic system? So I set out and I characterized this into MSCs. I varied each component of the circuit individually, holding everything else constant, looking at yield, fold change, homogeneity, et cetera, all of which is compressed in this heat map. But then I was able to, and I hope this is where you can see how much flow cytometry really made it possible for me to characterize this circuit, is iteratively I was able to improve this circuit from something that was really non-homogeneous with very poor dynamics and barely manufacturable, into just by reducing expression level in this case, we improved the homogeneity of the product from basically all off to at least bimodal. Then reducing just the affinity of the zinc finger increased the homogeneity to a nice single population and still gave me the inducibility. And then changing the internal elements of the cassette made it so that I could induce it fully, have a full population shift, and gave us the circuit performance that we wanted. Comparing that to CMV, which is one of the stronger promoters that we can use in MSCs, this viral promoter is usually our top end of what we can make the cells do on expression maximum, not necessarily expression homogeneity, because obviously there's a negative population here, but we outperformed the top promoter by 10x. When I show this and I change this to IL-10, which is a potent immunomodulatory cytokine that MSCs do not express at all, but is really commonly used by other regulatory cells, we were able to, again, over 10x the yield of the IL-10 that was produced from these cells with a significant dynamic range, which means I can tune the dose using this inducer. Then when I co-culture that with activated immune cells, we were able to just like decimate, frankly, the inflammation, but again, superior to CMV in every way in suppressing this inflammation. And this is where we got really excited. We previously had tested CMV expressing IL-10 in our model of traumatic brain injury. And again, I'm hiding a lot of really awesome data and flow cytometry and multiparametric analysis here. But CMV alone was able to suppress the activated microglia in the brain of these rats after inflammation. So if we look at this plot again, I'm very excited to see what this level of suppression can do in this animal model, perhaps resulting in a more potent therapy for traumatic brain injury and then long-term other indications. So to conclude here, my long-term goal is to take this technology, as well as others that I'm not talking about today, to build a universal platform or a universal chassis for putting cell therapies that are programmed with synthetic biology into the clinic. And I'm really grateful to my advisors, as well as to the funding sources that have made it possible for us to do this work so far, and the ongoing support specifically from REACH, from Mission Connect, One Small Step, and the Center for Translational Injury Research. So with that, I'll take any questions. Thank you.
That was another really cool talk and cool technology. My question is kind of about including the spacers. Is there value to that? Are there genes that are that big that you might want to be transducing?
Yeah, so every expression unit is about 2.5 kb by the time you include the full promoter, the open reading frame, and the terminator, if you want them to be independent. So this 2-gene cassette that we did was about 8 kb in size. The stuff that I'm working on in other projects is pushing 20 kb of cargo and 4 to 5 genes.
Are there, okay, so combining genes. Does that still have the same like detriment in sort of expression burden? Yeah, okay.
Yeah, so doing this project, I was trying to do multiple genes to change the surface profile of the cells using a single delivery. Now what I'm going to try to do is do 2 transposons together, which should overcome that deficit that we saw.
Thank you.
Yeah, question.
It's a very nice talk. So one of the things that we've seen putting transgenes into genomes of different cells, T cells, other cells, is that the promoters get silenced quite a bit. CMV is particularly bad at that. So I was just wondering, one, how long, what's the time course of your cellular therapies? And if they're longer, have you looked at passaging these cells as long as you can to see if these promoters do get silenced?
Yeah, I don't know if I can show you, scroll back that quickly, but I have no idea how to run that. Anyway, the data that I showed with the reproducibility, where I said that we took off selection, that's a CMV promoter. And so we went 4 more passages of fairly aggressive growth kinetics in order to, and we didn't see any real significant silencing there.
You see after about 2 months.
Yeah, so the MSCs, we can only go to passage 5 and then we freeze them. So we're not doing months of passaging. And they typically only reside in the patient for, max, really 24 to 48 hours after you infuse, but probably at most a week. So we haven't, this cell type specifically, it makes it so we don't have to worry about that, which is one of the reasons that we chose it. Anyway, thank you.